Hello, everyone, and welcome to today's webinar, Breaking Barriers for Biomarker Translation Toward Validation, Creating a New Verification Class Mass Spectrometer with the Stellar Mass Spectrometer. I'm Christina Jewell, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. Today's presentation is also interactive and we will have several polls for you. In fact, our first poll will begin now. So on your screen, you'll see a question. How many biomarker target analytes do you normally include in a study? You have 3 choices. I'll give you a few moments to make your choices, and we'll also be sharing the results for you. Again, how many biomarker target analytes do you normally include in a study? And as you answer this question, I'd like to now welcome our speakers. Dr. Philip Remeš, Senior Research Scientist, R&D, Thermo Fisher Scientific, and Dr. Deanna Plubell, Postdoctoral Research Fellow in the Michael J. McCross Laboratory at the University of Washington. For complete biographies on our speakers, please visit the Biography tab at the left of your screen. Dr. Remeš, you'll be going first. You may now begin your presentation.
Okay, thank you. Very happy to speak to you all today. And here we see on the poll that only one person, I think, has responded that they use between 100. No, here they come. We say most people are using between 100 and 500 biomarkers in their studies. So this is now, it's getting into the range where things can start to get tricky and manual work doesn't always cut it. And we want to talk to you about tools that we've made to help alleviate this process. So as you know, we released the Stellar Mass Spectrometer at ASMS, and there's 3 main pillars that underpin its performance. The first being like the basics, the hardware that we have 40 years of making mass spectrometers and ion traps for much of this time, and we have the fastest, most sensitive ion trap that we can make there. And so this is what underpins everything. But then we have some new things that are trying to help extend and make things easier and extend the scale of the studies that we can do. So we have an adaptive RT, which is a new way of acquiring targeted data that adjusts the targeted windows as the chromatography shifts. And we have some tools built into Skyline that help to create assays. We're going to look at all of those. There's, maybe broadly speaking, there's 2 worlds in the mass spectrometry. There's the discovery world where all these exciting things happen about almost getting to the whole sequencing a human proteome in an hour. And on the other side is the precision side, which is a much more conservative side, which is not interested in measuring the whole proteome in an hour and is much more interested in just very targeted experiments, measuring just a few proteins perhaps. And their goals and needs are different than the discovery world. And we're, with the Stellar, trying to bridge these 2 worlds by having a system that can do the targeted experiment that the clinical world is used to, but being able to scale it up to discovery scale. And the targeted experiment, as you know, is very simple. We have a list of targets that we're going to measure, and we simply say, what is its mass or charge? Maybe it's charge, which can help fragment it correctly. And then when we want the instrument to measure it, and we'll get plots like this, which in Skyline can be used to integrate the peak and tell us something about the abundance of the peak. And we could say prior to ASMS, I think, at least the way I see it, the targeted experiment hadn't really changed all that much. There's some things going on in the background, but still it's difficult to do. The coverage is kind of low, and it's not so easy to scale. Even to 100 targets starts to be a burden sometimes, although it is very sensitive and its throughput is very high and it's very reproducible. So it has these characteristics that, you know, the clinical world is interested in. We, with Stellar and with these new tools, we're trying to make it so that it's much easier to use. So you can make assays for just 100 or 10 or 1,000 targets, and so it's the scale is much higher. But no matter what the scale of your assay, there's a set of questions that you have to try to answer. There's a set of problems that you have to say, what are the compounds that are in the sample, which are possible to target? Which ones are worth targeting? Because now, unlike DIA, where you have some unbiased sampling and maybe you're wasting a little bit of the instrument resources, here we're going to focus the resources on things, but we have to know where to focus them. You have to answer the question of which collision energy to use, and I'm not going to get into that in this talk. You could ask me later, but we're using this normalized scale, which tries to make every system basically have the same performance, and we usually don't tune for our peptides, but one could. We have to answer the question, which transitions use for analysis, how many precursors could we possibly do? You have to know how fast the instrument is, and you have to get all that information into an instrument method, and then you have to maintain this assay with time. And broadly speaking, you could say that there's two kinds of targeted assays. There's the more traditional one that's more closer to what you have in the clinic, where you have internal standards for every target. And we won't talk much about that here, but we do have our bioarchives paper where we show how to do this with the PQ500 assay. But here we're going to be talking about how to take results from some like discovery run where there isn't an internal standard, and these assays can be sometimes much larger. As a basis for creating this assay, we can use any of our thermal mass spectrometers that have full scan capability. So you can use Stellar to create an assay, or you can use a Tribrid or an Astral or an Explorus. Any of these ones, we can take the data from that, put it into Skyline, and we can make an assay. And we like to use this procedure that was been popularized by the McCoss lab. I think in 2018 was this Searle paper in Nature Methods, where in order to make a nice, robust DIA method, the strategy was to first create a library very carefully with multiple injections, where each injection would just look at a certain segment of the precursor space. And then the idea being that later on with a DIA method that is going to look at, say, 400 to 1,000, anything that you could see in that assay would have already been characterized in the gas phase fractionation library. And Deanna showed with her preprint there that you could use that source of data, this gas phase fraction library, to create a selected reaction monitoring assay, like for a triple quad. And we're going to use that data also to make our targeted assays here for Stellar. And the general workflow is that we're going to get that data from whichever of our instruments. We're going to try to detect peptides in it. We usually use Proteome Discover because that's our software, and we use Chimeras, which is this new advanced algorithm for detecting peptides. And then we'll import that into Skyline, and we'll create a method, and we'll acquire that data, and we typically will then stream that data to Ardia as the data are being acquired, so that our hard drives don't eventually fill up. So now I'm going to start doing a sort of live demo of this software. We're going to look at 3 tools. 2 of them are kind of minor ones. We call them the gas phase fraction, the GPF creator, or the GPF importer. And then we're going to look at the PRM conductor. And in between those, the first 2 and the second one, we're going to look a little bit at some of the algorithms we use in PRM conductor. And these data and like a walkthrough can be found at this link here on Panorama Web. So in this first little demo, we're going to try to just make the methods. And this is something that's come up as, it's just the first place that someone can make a mistake, the first stumbling block, if you will, into making a targeted method, is that you have to make maybe six methods, 6 instrument methods, and each one you just have to change one parameter, but it's a way to make a mistake. So I will try to share my screen, and this should come up in a moment. I'm gonna select my other screen, and here I have a Skyline. I believe you're going to see it in one moment. Now you see it. Okay. So in Skyline has this very nice little ecosystem where they have a tool store, and you can install various tools. So I've installed our thermal one, and I can launch the GPF creator, and it should, and one of my here, it opened up in a different window. So I have to drag it over here. So you see this tool, what it's meant to do is just automate the creation of these methods. And this tool knows something about how fast the instrument is. And if I say that I am going to have an hour method, and maybe my peaks are 20 seconds, then this tool knows that what you should probably use 3 experiments to do that. So one will use a template method, and here is a template method that has, we usually use one that has 3 methods in it. One of them is for doing adaptive RT. It enables this this adaptive RT algorithm. So it is just collecting some data that's going to be used for alignment later on. Then, and the meat of the method is just, or of the experiment is this DIA one that has a precursor range, and it takes a little while to update, but here you see it's 450 to 550. So I've selected this template method and I say create the files, and now it's going to create 3 files, and they will eventually be put here. Everything goes a little bit slower, I guess, as we have things running. But so, okay, see, the first one was made, and it shows up in this window. So if I would open that file, it would just have this precursor mass range changed. If I can select the first one, and you'll see this precursor mass range will get updated to 400 to 600. So this is the first little tool that we have. So now I would like to show you the second one, and this is a tool for getting the data after we've acquired our gas phase fractionation library. It's a tool for getting the data into Skyline. And Skyline, of course, has a way to do this already, but it has it can be a little bit difficult because there's lots of settings, and they're, by and large, they're settings for high resolution accurate mass instruments, and Stellar is more like a triple quad in terms of its resolution. And so we'd seen that when users were trying to import these methods, they would do it wrong. So we just said, we'll make a little tool just to put the data into Skyline. And I also note that the Skyline team is working on doing like having different profiles you could use. You could have one for Stellar, you could have one for your Astral, for example, to import the data. So now if I have the tool launched, and what it does is we select the results from Proteome Discover, and those ones, they are in a file over here. With Proteome Discover, after from 3.1 and after, the results have, there's a PD result file, and then there's a result details file that has the spectra. So you need both to be in the folder, but you just select the result one, and then you can select the raw files that you want to import. And when you, let me see, so if I select these, they'll show up. And then if I import to Skyline, what you'll see is some outputs from Skyline. Is it just changing all these myriad settings that one would have to deal with? In a moment there, you'll see, yeah, so it's like changing the charge state, so just all the ones that we usually use. So this will set up everything for you. So now I'm going to unshare my screen here in a moment, and we're going to talk about some of the algorithms that we have in PRM Conductor before we launch that program. Okay, so I'm back, and you should be able to see my slides. Here I had the one that was talking about GPF importer and just saying that yeah, the Skyline team is working on improvements there. So in the future, you probably won't need that little tool. So let's talk about some of the algorithms in PRM Conductor that you're going to see that are helping us make assays. So Skyline already has a tool called the scheduling window. So you can look at this, and it will show where the different targets in your assay, where they're eluting, and how many are active at a particular time for a given scheduled window. So with the 1 minute window, we're saying for every target, we're going to try to acquire data for it in that, in plus or minus 0.5 minutes from where we think it's going to elute. And that depends on how comfortable you are. Traditionally, it depends on how comfortable you are with your chromatography shifts. If you're going to get 2 minute shifts, then you need maybe a 4 minute window. And it's a little bit difficult, or it always was for me, to try to know whether an assay, whether there are too many targets or not for my assay. So we tried to solve this with an algorithm, we call it load balancing. And there's 2 components to it. One is that we're going to look for every peptide, or sorry, for every protein in the assay, we're going to try to pick its best peptides. And the user has the ability to select how many, if it's 2 or 3 or 10. And then we're going to put them into the assay only if we have time to acquire the data for it. We call that load balancing. And you'll see also we have optimizations for making either very high throughput methods that could give up something in terms of sensitivity, or just like the default one, which is still very, very high throughput. So this first optimization is the load balancing and picking the N peptides per protein. So we take all of the proteins, we sort them, each one according to the peptides, the quality of them, and we're going to try to put them into the assay. So we take the first orange one and we look at this peptide and we see that it does, in fact, it elutes at a certain retention time, and the acquisition window width is given by the x-axis, and the y is saying how long it takes to, how long our estimate is that it would take to acquire data there. The next protein, then we go to it, and its best peptide, it elutes at a different part in the run, so there's no problem, there's no collision. Eventually, we'll get some peptides that elute at the same time as another, and we add them together, they accumulate, but it's still below this user-selected cycle time. Eventually, we could get to the situation where a peptide couldn't be added without going over that cycle time, and so we won't add it, but we would then consider its next best peptide, and it elutes at a different time, and so it now fits in there. And this now helps us pack in as many targets as we can while still respecting the cycle time. Yeah, so in the end, we'd have a fully completed assay. And there's two modes to use the Stellar, basically. If you're just talking about MS data, there's a conservative way, the default way, which is where every peptide would get at least a certain amount of injection time, say like 13 milliseconds. And then there's a more aggressive way where we can add in more peptides. Say if you want to push the chromatography to 10 minutes or thereabouts, and you still want to go after many peptides, you could do that. And so we're going to look at that next. So Stellar uses a quadrupole linear ion trap as its mass analyzer. And this mass analyzer, it scans everything out. So the amount of time that it takes to acquire data depends on the mass range. A typical peptide mass range is, say, from 200 to 1500. So at the low end, we just want to get at least like the Y2s. Those are the first informative fragments. And at the high end, the high end can kind of depend. Like the Kuhn lab had a paper where they said maybe you only need to acquire data up to 1100. We had seen that a general range might be up to 1500. But you see that it took us maybe 10.4 milliseconds to scan across this range. But this peptide, it only has transitions from 200 to 1000. So we could narrow that down and we could save 4 milliseconds. Those 4 milliseconds could really add up if there were hundreds of targets that were active at a time. And we can also see that, say, the only transitions that were good, that we are interested in, were, say, Y5, 7, and 8. So then we could acquire data just from 650 to 1000, and we could go even faster. So one could do that for every single peptide, but it would get pretty tedious to do it manually. So our tool, the PRM conductor, it does this for you. And I'll show you a little demo first before we get to the live demo, showing that this is now one of these concurrency plots, but it's our version that has the instrument time on the y-axis. And it's saying here we have like 4,000 total possible targets that we could go after. But because the cycle time in this horizontal dotted line was what it was, like 1.6 seconds, only 2,400 targets could be analyzed. If we press this optimize scan range button, though, we can acquire data much faster, probably around 100 hertz or so, depending on each peptide, and we could acquire data for 3,300 targets. So we can greatly expand the throughput by using this optimization. And we did this in our preprint on bioarchives for the assay with the PQ500, where there's 800 heavy standards, 800 endogenous peptides, so 1,600 peptides to try to target. And so we did that in it was a 14-minute method in the 100 SPD method. So we needed that optimization to do that, to scan really fast. Whereas for the 24-minute method, we could do them all without that. So, and we showed in the paper that you give up like 60% or so you gave up in the LOQ by going faster, but this is something that's understandable and easy to think about, the trade-off that you make. The final thing, little algorithmic thing we'll talk about before the rest of the demo, is that a user has to choose what is the acquisition segment width. Now, because of the adaptive RT algorithm that we have, we usually use about 3 to 4 peak widths. So if my peak width here is about 0.2 minutes, maybe we're going to use 0.6 or 0.8, or this one has a minute for acquisition segment width. And this is much narrower. It's like a factor 2 to 3 narrower than you might be comfortable with if you had to deal with the chromatography shifts. And again, here's a little demo showing that if you were going to use like a more conservative window size previously, say 3 minutes, then in this assay you could you could target 1,500 targets if you were, but it would take up to 9 seconds if we wanted to acquire data for all 4,000. If we narrow that down to 0.75, which is much more typical for this assay, for this chromatography length for Stellar with adaptive RT, then we could go to 3,700. So that's going to help us out a lot in terms of increasing the throughput. Or if you just had a few hundred of peptides, then this is going to give us more accumulation time for each one, giving us better sensitivity. I forgot there's one more thing, is just to introduce what is adaptive RT, because you'll see in the demo how we're automating the setup of the method for using adaptive RT. So this is a method that we've been working on for many years now, that is it really helps you to not have to worry about your assay over time in those chromatography shifts, because what we do is, in say a discovery run or a previous targeted run, we collect a set of data, a set of reference data, and we're going to compress this down, and we're going to include it later into instrument methods, and we're going to align against it. So in the next run, we will collect similar data to what was collected in the discovery run or the previous run, and we're going to do a cross correlation. We're going to compare it to every point in the control and get an estimate of how much the data, or how much the compounds are shifting relative to that reference. And that's what I said there. And then we're going to update the targeted schedule accordingly. So here we're going to have to shift everything by 2 minutes, from 32 to around 30. We have this nice example that we'd like from a collaboration we have with the Van Eyck lab, where we collected data for about a month, and we have a little GIF that's playing, that's showing these correlations. In light colors is the maxima, and the cyan line is showing where we think the maxima is, and the white dots are showing where Skyline thought the peaks were, and you see that they overlap. And the nice thing was that we changed columns at about 19 days. So as the video plays, you saw this huge jump, and that was where everything eluded differently, but we were able to see where they were based on the adaptive RT algorithm, and we can adjust the windows based on that. Okay, so now we're ready for this demo, and what I'm going to show you is how we refine the transitions, then how we can sort of live see how they fit into an assay, and then how we can create that assay. So I will try to share my screen again, find my mouse, and find the screen that I want to share, and it takes a second. Entire screen, and now you'll see it in a moment. Update. And there it goes. And I can see that it's going a little bit slowly, and I guess the running the webinar costs some computer resources. But you see down here on the bottom, Skyline is exporting it's exporting a report. That report is shown here. It's just a basic set of data and a set of metrics. We have things like the area, the height, the background. We're going to be taking an area to background ratio. It has something called the shape correlation, which is an important metric for figuring out if you have transitions. So that also came actually from that Searle paper that was referenced before, where you could look at all the transitions for a particular peptide, and you could figure out a median, something that's characteristic of that peptide, and other transitions that don't elute at the same time, or sorry, they don't have that same shape, they're going to get filtered out based on that. So this one is clearly from some other kind of peptide. Some of these other ones are kind of low, and those might be filtered based on other metrics. Right. And we also have things like, yeah, the whole chromatograms, we're eluding, we're sending those. So it does take a little while. So I think as that one loads, I'm going to go and talk about some of the other slides, and we'll go back and share in another moment. Usually it doesn't take that long. Usually it'll just take like 10 seconds for that one to load on my laptop. So I wanted to talk about some of the things that the instrument is doing to optimize the data quality, and to make sure that nothing bad happens, or that the data quality is as good as it can. And one thing that we've had for a long time is something called automatic gain control, or also known as AGC. And what we're trying to avoid is the situation where we put too many ions into the trap. Now, since 2003, we've had these traps that have a really big volume, and it's kind of hard to overfill them. But if you try really hard, you can, or if you go to really narrow chromatography, you could overfill. And you could see that in your Skyline data sometimes, where maybe they would drop out at the beginning or in the middle of the peak, and you might think it's spray stability, but if if one looks into it, in certain cases, it could be that you overfilled. So we want to avoid that. And oops, shoot. If the way that we do it is very simple, we just look at a previous acquisition. That one was acquired with a certain injection time, and a certain number of ions were measured. So that's a flux, that's a ratio, and we'll multiply that by the target, and that will give us the new injection time that we wanted. And we will use the device that we have in the instrument called a split gate to very precisely meter ions in for the amount of time that we want. And we can go down to close to a millisecond, or sorry, close to a microsecond. And what we'll do in the end is we'll measure a certain number of ions, we'll divide by the gating time, and that'll give us like a flux and intensity. So now we will. Okay, and now we're going to look at this video that is showing us how that process works. It's a special device called a double split gate, and I'll talk about why that is. First, we were blocking with the back. Then we're going to let in ions for a certain amount of time, say it's like a millisecond or so. It's probably just a few microseconds here. And then we're going to block with the front. And having these two sides of it helps us go down to these very narrow, like microsecond times. Now at the end, you saw all the ions get deflected into the front part of it. So this is an important part of the AGC process. Being able to go low helps us go to very high concentrations where you don't want, all that many ions. The flux is high, and this figure is trying to show you what the AGC process does for you. The dynamic range of concentrations that you can go after, because you have a certain range that's limited based on your detector. You can detect down to like a single ion, but maybe you actually need 10 or 20 or however many to get good statistics. So on the low end, you're limited by that. On the high end, you're limited by not saturating your detector. And you have this other range that is on the low end based on how low your split gate can go. And we have the double gate for that. And on the high end, by how fast your chromatography is and how many points per peak you need. So this process allows us to go to, say, like 8 orders of concentration, which is higher than what the electrospray process can bear. So the saturation is not a problem on that high end, because first, ESI is not going to be able to provide enough protons to ionize everything. And we also have a newer optimizations to the AGC process that help you not to have to adjust parameters. So previously one would have to worry about the maximum amount of time, the maximum injection time it was called. And if you were looking at just a few targets for very low concentrations, you'd have to go to a high time, or otherwise, if you had a lot of targets, you'd have to go to a low time. Here with this thing called dynamic, we adjust for you. So at the beginning of any cycle of acquiring targets on the instrument, we look at the targets we want to acquire. We estimate how long it takes us at a minimum to acquire that data, and then we compare that to how long the cycle is going to be. And in this case, it's we have this big space of extra time that we can divide up into the targets. In this case, now I've depicted the low concentration ones as red and the high concentration ones as white. And based on how they've expanded, that's how we've distributed this extra time to them. So the low concentration ones, they got more time because they need more time to try to get good signal to noise in the spectrum. And so now we just, by default, we always use the dynamic setting. And I was talking about maybe the one place where you could still might be possible to overfill a trap even with a big capacity might be with very, very narrow chromatography, where the flux would be changing so fast that we wouldn't have time. Based on the previous scan, we might think that we had a low flux of ions, but the next spectrum, it might have a high one. So what we've done is, and we described this in a talk a couple years ago at ASMS, we've trained a neural network to know what is the shape of typical LC peaks. And we've done this for a big library of different peak shapes. And so in real time, we collect the data, we collect a set of histories, intensities for every precursor. We pass it over to the instrument computer where we have the neural network sitting, and we predict what is the next intensity, and then we use that for setting the next injection time. And this slide shows you a few of the different peak shapes that were used in our library, from very narrow, like 1 second peaks with capillary electrophoresis, to different gradient lengths, then up to things that look like ion optic columns that have a lot of fronting and tailing on them, which are very difficult actually because of how fast the intensity changes at the beginning. Okay, so now at this point, I can show you I can do the part of the demo with the PRM conductor, because that is loaded up. And I'll share my screen again, and I will put over there. Now, yeah, good, you can see it. This is the PRM conductor program, and it has maybe three main parts. The first part is all about filtering transitions based on these metrics that were exported in that report that I was showing you. There's the area, there's the signal to background, the relative area, this time correlation that I was telling you about, the shape, and then how wide the peak is. And if you update any of these, you could update it, and now eventually it will update, and the vertical line will move to the left, and the number of precursors and number of transitions that have been filtered would change a little bit. And you see that there's 99% of them, almost all the transitions are passing this filter. It's not a very heavy filter, but over here, 42% of the transitions are passing, so 60% or so are getting filtered. So there's those parameters. Then there's the important one. There's how many good transitions are we saying, or how many transitions are we requiring the precursor to have in order to be considered good. So here's a distribution here saying if we required only 2, then we could gain a few more precursors. Most of them have quite a few good transitions actually. And 2,700 of our 7,400 total, these were E. coli peptides that were detected, could be possibly scheduled. Now in the second part, we have this defined method part. And this one is neat because it's telling us something about how fast the assay could be acquired. So it's saying that we need at the maximum to acquire all 2,700, we would need 4 seconds at the maximum. But if we had an Astral, for example, and we're acquiring data at 200 hertz, then we'd be able to acquire data for all of these. Eventually it'll update there, and we would see this thing that now it only takes a second or so to acquire those data. And we can do this for any of the parameters, and we can see how things change. Now, if I click on this button over here that's called balance load, we're going to see the plots that look more like what I showed you before, where we just have a single assay is being, before if I unclick this, then we're saying we're going to export data for 3 total assays. We want every single one we're going to acquire in separate assays, but I could just say I want to make the best assay possible. We could say that I only want to have the top 2 per the top 2 peptides per protein, and this would give us more accumulation time. You see, dynamic AGC at this point in the run would give us more injection time. We'd have higher quality data, or we could say, no, I actually want the biggest method possible, so I'll just make this a big number. And then what we'll do now is we'll show how we can export these data. Oh, I do want to show you that you could change to MS3, and this would cut the number of targets you could have by about in half or so. So we're going to select a method template and export the method and see what that did. So this is the targeted template. We're going to export the files. This will just take a moment, and we can try to load it up. This targeted file that was made, I hope, did I go? The targeted file that was, oh, it's still exporting because it's so slow as we're running. What's made is going to look a lot like the template for the GPF. It's going to have an adaptive RT experiment. It's going to have a targeted experiment instead of the DIA one. There, it finally went, and we see this one called demo. And when it loads up, we'll see that different parameters that from the PRM conductor have been imported for you. So the chromatographic peak width will have changed to whatever it was in the settings. The cycle time will have been changed, and all of those targets, those like 1,600 or so, will have been put into the targeted table. And when it finally loads up, I also want to show you that we've been able to take that data from that GPF, and we've embedded all those spectra into this little file, and you'll just see it based on this a little setting in the adaptive RT section that says, yes, we've imported that data, and now we can use it for aligning in real time. And this, unfortunately, is taking a long time, but it may happen in a second. It finally came. And so I can select the TMSN. And I will show you, yes, down at the bottom. Well, you see that the cycle time was changed to 1.68. We have 7 points per peak. We have the scan range could be defined with the optimized scan range. That's why it's in the table now. So we just use 200 to 1,500, but it could have been customized for each one. And This thing is going a little bit slow, but down at the bottom, you'd see that the adaptive RT section is filled out. It says embedded file, and we'd be ready to go. So let's stop sharing, and we'll go to my conclusion slide, which is just summarizing the fact that we have this program called PRM Conductor that's in Skyline. It helps us make methods, whether they're big or they're small. It helps us eliminate the usage of scripting in Excel. We have a new hardware that enables everything, and we have a new algorithm for acquiring data called adaptive RT. So with that, I'll be passing it over to Deanna then in a moment.
Thank you, Philip. And I'm going to bring Deanna back up on here. But before we dive into Diana's presentation, we have one more poll for our audience. What is the most important metric in your biomarker experiments? We have 3 choices. So we ask our audience to participate in this. Just select one of the 3 options. Again, what is the most important metric in your biomarker experiments? We're going to share our results with you here. So we love your participation. Okay, they're coming on. And then Deanna, we'll go ahead and bring you on back up to the screen here. Thank you, Philip. All right. Deanna, you may now begin your presentation.
Sounds good. I'm excited to get to share with you guys today some of the work that we've done now constructing assays using this new instrument, kind of for a range of different applications or processes. So just to start off, I want to kind of, highlight and remind everyone kind of when we're thinking about the data that Philip's kind of talked about already, putting it into context of maybe what we've kind of classically thought of when we're doing targeted methods, specifically targeted like proteomics methods. In a typical triple quad method for selective reaction monitoring, we are selecting which precursors we're wanting to monitor before we, build the method. And then we also, from that precursor, select which product ions we specifically want to monitor as well in that assay. And of course, we can monitor several different product ions, but we have to know that kind of ahead of time and know that we are targeting the specific acquisition of those product ions. So parallel reaction monitoring methods kind of cut out one of those steps. We still have to select which precursors we're wanting to isolate before, we acquire the data. But when we actually acquire the data, we're acquiring this full spectrum. And then we can post-acquisition do a targeted extraction of that data to end up with our kind of base unit, which is this chromatographic peak. So this is tracking as these product ions elute off the chromatography. We get this nice peak shape. And this is just extracting all of the charge 1, Y, and B ions for a peptide. And you can see we can kind of get information about kind of the area under the curve that we can use for quantitation. And just to remind you that like each point on this peak is a different full scan. So if you kind of march through here, you can see the signal increases as the peak kind of reaches its apex and then slowly decreases as well. So this makes it kind of easy to then, do some things after acquisition to improve our quantitation. We can select a subset of ions that we think are going to be the most reproducible or give us the best signal and things like that. So fundamentally, the only thing we really need to build a really simple PRM method is just a list of precursors if we aren't concerned about scheduling for a small sample of things. And then we provide that precursor list to the instrument and can then do the data analysis. In our case, we do it in Skyline, where we do the chromatogram extraction and then quantitation. So I'm going to talk about the kind of a handful of different assays and specifically go into 3 separate ones, but kind of highlighting the kind of range that we've constructed using this instrument. And the first one that I'll talk about is this retinol binding protein assay. That is a very small assay that we didn't need to do scheduling because we're only monitoring a handful of things. So kind of like analogous to what we had previously do with the triple quad SRM assay. So this actually relies on previous work from Phipps et al. In the Hufnagel lab, and it's specifically for retinol binding protein in neonate plasma. So retinol binding protein is a marker of vitamin A sufficiency, and it's commonly measured in clinic by nephthalolometry assays, but those require a amount of sample volume that can sometimes be challenging to get from neonate plasma. So instead, what they produced was a triple quad assay that enables us to measure a smaller volume to calculate the concentrations in. So it relies on monitoring and doing quantitation on 2 tryptic peptides that are derived from this protein, and use heavy labeled peptides as internal standards for the concentration calculation. So basically, we just wanted to recapitulate that assay and kind of compare and contrast building an assay in this new instrument by PRM, comparing it to a triple quad assay that they use in clinic. So we already have information about which precursors they are targeting in their previous assay, and makes it really easy to build a method, in this case, an unscheduled method. And I'll actually point out that this is that specific peptide that I previously highlighted is one of the ones that we're monitoring. So importantly, we want to test whether we get the same results or not. So here I'm comparing measurements done on a range of samples that were previously measured by the triple quad SRM method, and then those that we measured by the PRM method. And overall, I think you can appreciate that we're pretty much recapitulating the results very closely. So that's kind of a nice positive thing to see. Now, of course, the nice thing with this instrument is that we can also target a lot more things concurrently, like Philip kind of mentioned. So we can broaden the scope of this assay potentially in case that was kind of interest. So here are a couple additional proteins that I added as targets, specifically kind of highlighting the transthyretin protein, which we know is important in relation to retinol binding protein because it actually occurs bound with vitamin A in circulation as well. So we can easily just add some additional precursor targets to our list and come up with a slightly larger assay, and still very broad scheduling, not very specific. Of course, we still want to make sure that we're measuring the same protein just as accurately when we're including additional targets. And we can see that when we include additional targets, we still get a relatively close, relatively reproduced values. But of course, in addition to that one thing that we are monitoring, we have information about additional targets, which could be potentially helpful. Of course, that's pretty basic. We can go through all of the individual data manually if we wanted to in Skyline and filter out anything that had interference or things that we wanted to remove from our data set. But when we get to larger and larger assays, it's much more challenging to, first of all, come up with which precursors we even want to monitor in the first place, and then also come up with which transitions we then want to use for quantitation and know which ones will provide us the best information. So in thinking about doing this, one thing that we've done in the lab that Philip kind of mentioned is we have previously made use of prior knowledge that we gained from these gas phase fractionated DIA libraries. So this historically was done when we were constructing triple quad SRM assays, and we made use of data that was acquired on an Orbitrap mass spec doing these narrow window DIA runs. We then extract this information in Skyline, where we can then use different sort of cutoffs or filters to refine our list of things that we think are going to be the most reproducible, and then also select the transitions that we think will perform the best in our assay. And that then can be scheduled based on the relative retention time that we actually get also from the data-dependent acquisition data. So when we've done this previously, it's kind of expedites that process of building an assay, and we can get really reproducible assays kind of on our first shot. So this is, again, only for 78 precursors by triple quad in cerebrospinal fluid. But overall, good reproducibility for kind of a first step. If we think about kind of the overall construction or like architecture of our triple quad methods, we kind of have these two different axes that we think about. The first is the retention time, so when they're eluting off of the column, and then the second is the precursor mass to charge. So here we can see that when we have these historically SRM triple quad measurements, we had pretty wide windows in which we were acquiring for each precursor in order to not kind of miss them if we had shifts in our chromatography. So that really reduced the number of things that we could monitor. So now we want to kind of expand this kind of framework for the new instrument and still make use of this prior knowledge that we can gain from these data-independent acquisition experiments, either on, like Philip mentioned, either on these high-resolution accurate mass instruments or on the Stellar itself, and then still make use of Skyline to extract and select and filter our data, but now with this PRM conductor tool that Philip just went through. And then we end up with a PRM assay that also uses this real-time alignment or the adaptive RT to kind of generate these really large assays. So this is, again, we're still needing information about which precursors we're targeting, but we're getting some of that information from this prior knowledge. Like Philip mentioned, just to reiterate, this is really where we're doing the majority of our method building or our method development. We're putting our data into PRM conductor and adjusting the parameters to kind of build an assay that is the best fit for what we're using it for. So we can do things like adjusting the dwell time and the scan range and stuff to kind of optimize for our specific sample matrix of interest. Again, we make use of this adaptive RT, and that really allows us to really narrowly schedule the acquisition for each precursor, and that allows us to kind of maximize the number of things that we then measure concurrently. So here I am showing kind of like the same plot from the PRM conductor, but for an assay in cerebral spinal fluid, showing across the retention time space how many concurrent precursors are being measured at any given time. You can see we're usually not really ever, or in this case, we're never going over 100 concurrent precursors at once, and that's going to help us with our cycle time and making sure that we're still getting good measurements for every target that we're wanting to monitor. We can then again think about kind of the architecture of this assay, both in the retention time space and the precursor mass to charge space. We can see that we're really covering a larger kind of swath of the precursors and doing kind of maximizing the measurements through the whole entire chromatographic space as well. So just to kind of put this in context, if we're thinking about kind of previous targeted assays that were, much fewer precursors, much longer windows, now we're able to generate these assays that have much shorter windows, and therefore we can target a lot more precursors, which also is making use of what Philip described of kind of optimizing the placement of precursors too, so that we're kind of not wasting our instrument time. Now, of course, we can build assays as large as we want, but at the end of the day, like you guys just answered in your feedback poll, we really care if it's quantitative or not. So what we want to do is kind of assess how well these large-scale assays perform quantitatively. Now, one way that we've done this in the past is using matrix match calibration curves, specifically when we're doing kind of these more discovery style, like DIA style experiments. And this allows us to get a relative limit of detection or limit of quantitation and calibration curve for every peptide that we are detecting or measuring in that experiment. So we'll use the same strategy, but now with these much larger parallel reaction monitoring assays. So here are two different assay sizes from the same sample source or same matrix, which is a human plasma enriched with magnet. And then we can compare things like how reproducible overall our assay is. So this is a distribution for all of the precursors that we're monitoring in an assay, their reproducibility by coefficient of variation across replicates at different dilution points into the background matrix. And you can see in 100% dilution, we have really reproducible measurements. And as it gets more diluted into the background, it starts to shift the distribution. But overall, we still stay pretty reproducible down to a pretty low percent dilution. We can also look at the accuracy of those measurements. So looking at whether or not the measured intensity in relation to the 100%, whether the dilution compared to the 100% is the expected ratio. And so here we can see that for the different dilutions, overall, they tend to kind of have a distribution around the expected ratio. But with a slightly smaller assay, we do see that we have a little bit better performance when we get into lower dilutions. So there is a little bit of a trade-off. If we go to really large assays, we're going to spend a little bit less time, and we tend to have slightly less reproducible or less accurate measurements at a global level. But you can see there are still going to be a lot of specific targets that have good reproducibility or accuracy. Okay, so we think that overall these assays can be pretty large. So now I want to give two vignettes of how we've used these large assays. And the first is looking at an Alzheimer's disease protein panel. So making use of this kind of workflow. But now we're also going to inform our selection based on previous literature and experiments. So this is based on two prior manuscripts looking at Alzheimer's disease and also preliminary data from our own study and looking at proteins that are differential in two of the three different studies. So we generate an assay that's monitoring 100 proteins and a bit over 1,000 precursors. Here I'm showing you kind of where those proteins fall in the dynamic range of cerebral spinal fluid. So kind of spanning the whole range. And then we can apply it to a small Alzheimer's disease cohort. So this has both Alzheimer's disease, healthy controls, and then Parkinson's with and without dementia as well. We can look at, since we know that these things have been found before, it's kind of nice that even in a really small cohort, we can still see a lot of the things that we expect to see changing in Alzheimer's disease. Things like you can see way up here at the top, there's tau protein, things like ApoE, and this A4 is amyloid precursor protein, which gives rise to amyloid beta. We can also see this really extreme outlier in ApoE E4 allele-specific peptide. And if we dig into the data and separate it by group, we can see that it's enriched in Alzheimer's disease dementia. We know that in the general public, Alzheimer's disease dementia individuals tend to have an enrichment for this E4 allele. And if we look in our data set, we can see that we do indeed have this enrichment in this E4 allele in our data. So then if we instead look at this peptide separated by the genotype of the individuals, we can see that it really nicely recapitulates the known genotype of the E4 allele, but whether it's heterozygous or homozygous. So that's kind of a nice way of like building a panel assay pretty easily to then kind of test to see if we can see the same differences in different cohorts. The next is this larger assay. We're calling it kind of a survey assay, and then we'll apply it in plasma. So again, using the exact same workflow, but this time we're only selecting things based on their kind of inherent properties. So we're not going into it with a specific hypothesis or a list of things that we know we want to measure. Instead, we're going in a little bit more agnostically and saying, I just want to make an assay that measures the things that are going to be the most reproducible or have the best signal, and as many as possible. So in following this method, we now, instead of 100 proteins, we're monitoring 700, which is a lot for a targeted assay, and a little over 2,000 precursors. And this is in magnet-enriched plasma, which is kind of previously described from our lab. But in using these methods, we can still look in to see how reproducible overall this assay is. So we have these quality control samples that we prepare and replicate. We can see how reproducible the overall assay is between them, which looks pretty good at kind of the global scale for 2,000, a little over 2,000 precursors. Again, we can apply this to a small cohort. This is also a small dimension cohort, slightly better balanced than the CSF one, but this is plasma. And even though we didn't go into it with a specific list of proteins or precursors that we knew we wanted to monitor for this disease, we can still find differences in these proteins that we can measure really reproducibly. So this we can kind of treat like normal discovery style proteomics to an extent, but in a much more targeted way, where we're really only targeting things that are going to give us really good reproducible results. So just to kind of highlight, we've constructed kind of a range of assays, and these really large assays are we think, pretty useful and powerful. And I will point out that, the concurrent number of precursors and the stuff that's going on when we're building our assay in PRM conductor really makes a big difference and really helps to make these assays as large and as quantitative as hopefully we can reach. So a couple key takeaways hopefully from my presentation. First of all, the full scan PRM really helps with kind of simplifying assay development. We only really need to know the precursors we want to monitor, and so that kind of eliminates some of the upfront stress or work. We can also leverage DIA data for figuring out what things we want to target and which transitions we want to extract based on kind of their previous results. PRM conductor is what we use to really easily generate these really large methods. And then also this adaptive RT is what also makes these really large methods possible. So these were all things that were covered in more depth in Philip's presentation, but this is kind of how we're using all of them to generate these assays. So with that, I think I will skip to this last poll. And yeah, thank you guys for your attention.
Your excellent presentation. Just realized I was muted. Thank you both for your excellent presentations. And now it is time for our live Q&A portion of our webinar. But before we dive into that, we have one final question for our audience. How do you currently verify putative biomarker candidates? You have three options. And again, how do you currently verify putative biomarker candidates? I'm going to give you all opportunities to answer that. And so if you haven't submitted any questions yet, please do so now. Just type them into the ask question box, click submit, and we will answer as many of your questions as we have time for. We are nearing the top of the hour, so I'm going to try to squeeze in as many as I can here. Okay, so let's just dive into this one. I'm going to ask you both this question, and Dr. Remeš, I'm going to ask you to answer first. How narrow can you set the scheduled RT windows without missing data?
So we typically, we were showing that typically like 3 to 4 peak widths is kind of what we recommend, but this adaptive RT algorithm that we have, it's kind of global in scope. So if you look at those cross correlation plots we had, the peptides, they don't all shift the same. There's kind of like a spread of them. So it's about a peak width or so extra. So we've gone as low as like 0.3 for like a very short assay, less than 10 minutes, and maybe up around a minute. But I don't remember what you were using, Deanna, for your assays.
It kind of ranged. I actually went all the way up to 1.5 minutes, but I actually found that I kind of preferred the narrower windows because it made like the peak integration and some of that more streamlined and easier to kind of look through the data without getting distracted by other spurious kind of peaks. But
yeah, we forgot to mention that, how it helps with data analysis later, that there's no other peaks, extraneous ones to pick. Yeah.
Excellent. Thank you. And Deanna, I'm going to go to you for this question. How does low resolution PRM data suffer from background interference?
Good question. I think all mass spec data suffers from some background interference inherently due to like background matrix effects. But I think kind of the benefit over like an SRM assay that may also have background is you don't have to necessarily know going in which specific transitions do and don't have background. Usually, even though a couple will have background interference, you can still have some that are interference free. And so you can still get a useful measure from that detection. So, yeah.
Thank you. And Dr. Remeš, we have time for one more question, and I'm going to wrap with this one. How does PRM sensitivity compare to MRM sensitivity?
So there, what you have to consider is whether you're looking at really big scale or really small scale. So if you go to just a few targets, then there's really no difference. We've done the comparison many times with our own triple quads, and like the limits of quantitation that you get with like our Altis, our top of the line instrument, are the same if you just have a few compounds and a few transitions per compound. But as you start to scale, then that's where we see bigger differences. And we've highlighted that in our bioarchives paper, that if you're pushing the triple quads into this range where they have dwell times like a millisecond or less, then they start to suffer. And then the advantage over PRM or like Stellar is really high and can be as high as like 10x for the limits of quantification.
Very good. Thank you. Now, I know we went through a lot of information today. Do either of you have any final comments for our audience?
No, feel free to get in touch with us somehow if you have questions, or hopefully this will be online later, I guess, for others as well to see.
It actually will be, yes. Today's webinar will be available for on-demand viewing, unlimited on-demand viewing, and in about 30 minutes or so, we will send you an email letting you know it's available for replay. So we encourage you to share that email with your colleagues and your friends who may have missed today's live event. Thank you again to Dr. Remeš and Dr. Plubell for their time today and their important research. We do want to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. I would be remiss if I didn't thank our audience for joining us as well. And we thank you for your questions. We had quite a few questions we weren't able to answer. And additionally, those that come in during the on-demand period, they will be addressed by our speakers via the email address you provided at the time of registration. So don't fret, your questions will get answered. And if you have any more questions, feel free to submit those and we'll get back to you. Thank you again, everyone, for joining us. Again, Dr. Remeš, Dr. Plubell, thank you for joining us. Take care, everyone, and have a great day.